Hello, everyone. It is my great pleasure to introduce today's speaker. Itay is an assistant professor at the Faculty of Life Science at the Bar-Ilan University, and he obtained his PhD at the Weizmann Institute of Science at the Department of Molecular Genetics, where he studied autophagy. Subsequently, he joined Steve Ellich's lab at HMS in Boston, where he really pushed high-throughput methods to really dissect the C and N-terminal rules for protein degradations in very innovative methods. In 2019, he set up his own lab in Bar-Ilan University, where he employs the genome-wide approaches and genetic tools to explore the ubiquitin proteasome system. And his research is really focusing on mechanisms that regulate substrate recognition and the implications for signal transductions and protein quality control pathways. Thank you very much for accepting our invitation today, and we are very much looking forward to your seminar. The floor is yours.
Thank you so much, Mikolas, for the very nice introduction and for the invitation. So, as Mikolas said, we work on protein degradation mechanism and their implication in quality control, pathway, and signaling. And you can guess from the title of the talk today that I'm going to talk about both ubiquitin-dependent and independent mechanisms of degradation. So, we all know that the ubiquitin degradation mechanism is only one way to degrade proteins. So in canonical mechanism of degradation, the ubiquitin chains that are built on the substrate serve as the targeting signal for the proteasome, while in non-canonical mechanisms, substrates are degraded in the proteasome in a ubiquitin-independent manner. And this pathway is less explored, and I hope to shed some more light today in my talk. So we will start with the Ubiquitin-independent project, but first I will try to explain the rationale behind our project. So normally, substrate in the ubiquitin proteasome system are targeted by E3 ligases that specifically recognize their substrate by interacting directly with degron sequences. And then when there is a close proximity, the E3 ligase can ubiquitinate the substrate and promotes its degradation. And when we look at how many E3 ligases are in human, there are around 600. However, there are very few degrons that were identified over the years. So when I joined the lab, we searched for some systematic method to develop in order to identify degron and match them with the E3 ligase that regulates them in order to better understand the specificity of the ubiquitin proteasome system. So in my talk today, I will show you what kind of approaches we use in our lab in order to investigate the specificity of the UPS. And then I show you some data from ubiquitin-independent degradation mechanism that we discovered in the recent years. And if we will have time, I will end up with ubiquitin-dependent degradation mechanism. And there I will dive a little bit more into the biology. So, okay, so let's start with the technology that we develop in order to identify degron in a high-throughput method. So this technology is named the GPS-peptidome, which stands for Global Protein Stability. So it's a method to monitor protein stability in a systematic manner. So the way it works is that a lentiviral vector encodes for two fluorescence proteins, DS-RED and GFP. DS-RED serve as the reference for the expression. And because these two proteins are translated from the same mRNA, then the GFP fused protein serve as the readout for the expression of the protein, the fusion protein, and the ratio between GFP to DS-RED reflects the stability of the protein. And the technology was developed at the Ellich's lab by Sherry Yan, and then other labs use similar approaches, including the Ebert and also some labs in East. So I've seen many variations of this technology now available also in other labs, and I heard that some other labs gave it some other cool name like the watermelon technology. But basically, previously people used this technology in order to look on full-length protein substrate and their stability. So they use manipulations such as different treatments or knockout of different E3 ligases, and then they assign the E3 ligase with the full-length protein substrate. But I remind you that the whole idea that we wanted this technology to work is to identify degrons. And we all know that degrons are short, and identifying a degron within a full-length protein might be challenging. So we decided, instead of using full-length proteins to GFP, to use peptides, libraries of peptides that originate from human proteins. So where is a good place to search for degrons? So we all know that the termini of protein are more disordered than in internal regions. And that's why we decided to first focus on degrons in the protein termini. And in evidence for that degrons are enriching termini is that actually the first degron that were identified in this seminal work from Barzalski lab are N-terminal degrons. And N-terminal degrons have been investigated for many, many years. However, for some reason, the C-terminus was ignored. And I just show you the C-terminus is as disordered as the N-terminus. So we hypothesized that there might be also degron in the C-terminus, but we had to find a way to find them. So we decided to synthesize a library of peptide that encodes for the last 23 amino acids of all human proteins, 24,000 different sequences at total, and then search for degrons. So the way the experiment works is that we synthesize the library, and then we cloned it into the GPS vector. So now the vector encodes GFP fused peptide in its T-terminus. And because this is a lentiviral vector, we make virus and infect cell, making sure that there is only one copy per cell. So each cell expresses now one GFP fused peptides. And then, because these are fluorescence protein, we use FACS in order to separate the different cells based on the stability of their GFP fused peptides. So you can see here, FACS plot. On the x-axis is the DS-red signal, y-axis the GFP signal, and we can see that there is at least a 2 order of magnitude difference between the most unstable peptide in bin1, where are the most potent degrons, and the most stable peptides in bin4. So we use FAC to separate the different populations, and then we sequence them. And then based on the sequencing read across the different bins for each one of the peptide, we can assign a stability score for this peptide that we name protein stability index for each one of the peptides. So basically, when we end up the experiment, we have an Excel sheet with 24,000 different sequences and their stability score. And now you can imagine what we can do with it. We can knock out E3 ligases and search for degrons that are regulated by specific E3 ligase, or change the condition and treat with different stresses and then look for degrons that are stress dependent. And what we did in the last few years is that since the development of the technology is that once we have degron in hand, then we want to find and map the critical residues in this degron, because it's still libraries of 20 amino acids. The degron can be anywhere in this along this region. So we use mutagenesis library. So now we mutate each one of the amino acid to the all other 19 possible amino acid, so we can find the critical residues in this degron. So that's one thing that we do. But then I will remind you, the whole idea of this systematic method is to match degron with E3 ligases. So then we do CRISPR screen with an E3 ligase library to find which E3 ligase regulates the degron of interest. And finally, the whole idea is to understand the molecular mechanism of recognition. So we want to understand how specificity is achieved. And for this, we collaborate with Chao Xu, a structural biologist, and then we can really define the molecular mechanism of recognition. So all together, all this pipeline helped us to really understand better degrons and how they are regulated. But we are interested more now about the biology. So we have a lot of information of degron E3 ligase pairs. Now we dive a little bit more into biology. So now in the first talk, I will talk about in the first part of the talk, I will talk about ubiquitin-independent degradation. And So basically, this project started where our original paper about the GPS-peptidome project published. So we summarized the whole C-terminal degrons that we were able to identify using this technology. You can see in this scheme, there are 11 different classes of C-terminal degrons. And we were able to map 8 of them to Kalin ring ligases. So Kalin 2 and Kalin 4, with their specific substrate receptors, were able to recognize this specific motif. The paper that I indicated here are on paper that look on the structural insight about recognition of the substrate. But then if you look carefully at the bottom, there are like 3 different classes of degrons. We know that these critical amino acids are important for degradation. If we mutate them, the peptide is not degraded anymore. So there were some motifs that are enriching alanine or valine, and we didn't know what's the E3 because at that time we didn't search for this E3. So the first project, when I joined Bar-Ilan University and I opened my lab in 2019, was let's find the E3 ligase that regulates those degrons. Okay, and then we do a CRISPR screen, like I showed you before, and we search for the E3 ligase, but unfortunately, those CRISPR screens fail. And there were 2 hypotheses that we thought, why should it fail? Because I told you, like 8 out of the 11 different C-terminal degrons were identified using CRISPR screen, but now we fail to identify those E3 ligases. So one option is that there is a redundancy. So there is more than one E3 ligase regulating this degron. So if we use our E3 CRISPR library and each one there is a knockout for one specific E3 ligase, there is compensation and redundancy. We won't be able to recover those E3 ligases that are redundant. Or the other option is there are no E3s. So it was easier to assay the other option, whether there are no E3s at all. But it was challenging to do it before 2018. And why? Because if we want to show that there is no E3 and no ubiquitination, we need to inhibit complete ubiquitination in cell. And prior to 2018, there were a drug that wasn't so very effective in inhibiting the E1. And there was thermosensitive E1 cells that weren't also very effective. And siRNA depletion of E1 didn't work well. Although we were able to reduce the protein by 90%, there was still very efficient ubiquitination in cell. And then we were lucky that in 2018, the great MLN7243 or TAC243 drug came out to the market, and we were able now in a very efficient way to inhibit ubiquitination in cells. So up to 1 micromolar for a very few hours, you see complete disappearance of the ubiquitin chain in cells. So we say, okay, now we have a great drug in hands. We can inhibit the E1. So how the experiment is going to work? We are going to look on substrate that are specifically stabilized in the proteasome. So in that case, both classes, ubiquitin-dependent and independent, should get stabilized by proteasome inhibitor. However, if we now treat the cells with the E1 inhibitor, only the ubiquitin-dependent substrate gets stabilized, but the ubiquitin-independent substrate should get degraded under the same settings. So we did some quality control checks. We took some peptides, C-terminal peptides, and then tested them with the drug. Here is an example of ubiquitin-dependent substrate. You treat it with the E1 inhibitor or with the proteasome inhibitor. You see a shift to the right indicating this peptide gets stabilized. However, now we take the 3 examples that I showed you just before that failed to recover any E3 ligase in the screen, and now you can see a different picture. We treat with the proteasome inhibitor, they nicely get stabilized. However, the E1 inhibitor doesn't do anything. And in fact, in some cases, you even see an increased degradation, enhanced degradation. We think it's because when you inhibit now the ubiquitination, so proteasome are less occupied, so they can degrade even more efficiently the ubiquitin-independent substrate. So that was the first indication that these substrates are ubiquitin degraded in a ubiquitin-independent manner. Of course, we did some other tests. We pulled down those peptides. We showed that there are no ubiquitin changes on them. So we did some other experiments. So we believe that these are ubiquitin-independent substrates, but these are just 3 examples that we had from the previous paper. We wanted to do something more systematic. So then we asked, okay, what is the scope of substrates? And what is the mechanism of their targeting? So in order to answer this, we use the peptide library that I just showed you before, this to better understand the scope of substrate and the specificity of the pathway. And we also wanted to find real full-length protein substrate of the pathway. And for this, we used the GPS Orpheum collection that we also have in the lab. So I'll start with the peptide screens. So we took the whole library of the peptidome. In that case, we worked with the C-terminal peptidome library. And then we treated them with the proteasome inhibitor or the E1 inhibitor. And we look for those peptides that are stabilized with the proteasome inhibitor, but not with the E1 inhibitor. And surprisingly, we find many, many examples. So before, if you read the literature about ubiquitin-independent degradation, you can find only a handful of examples of proteins that are degraded by this manner. But now we see that in terms of peptide, with a kind of artificial way of looking on this mechanism, you can see that there is pretty much the same number of peptides that can be degraded in ubiquitin-dependent or independent mechanisms. So the cell has the capacity to degrade ubiquitin-independent substrate, but probably doesn't see them much unless something really bad happens, kind of stress that open the structure of the protein, or other means where you need to degrade proteins in a fast manner and you don't have time to ubiquitinate them. And now we look on the motif. Okay, what is enriched among substrates that are degraded in a ubiquitin-independent manner? So here you look at heat map, where we look on the 23 amino acid peptide, stop codon here, and then the 23 amino acid peptide. And we look in each one of the position about the composition of the amino acid of the substrate that are ubiquitin-independent versus the ubiquitin-dependent. So if there are motifs that are enriched, you will see them in red, and no change or depletion in blue. And you can see that at the very C-terminal end, we can see enrichment of the same motif that I showed you before that we found in our original paper. So we have some alanine motif at the very end, we have valine motif, and we have cysteine motif. So specific amino acid can target protein for ubiquitin-independent degradation, but we needed to prove that indeed these are the critical residues. So here I just show 2 examples with our saturation mutagenesis approach. So for each peptide, we now mutate each one of the amino acid in the wild-type peptide that are indicated at the top to the whole 19 other possible amino acids. So we don't only say what are the critical residues, but what are the preferred residues in this type of degradation. And these are just 2 examples of alanine terminating peptides. So you can see alanine is critical for degradation in that case, and it cannot be replaced by any other amino acid. So you can see the motif is very short. At the extreme C-terminus is composed of only a few amino acid longer. So now we better understand what is the motif, but now we want to dive in and understand the targeting mechanism. So how those substrates get degraded in the proteasome. So if you look at the literature, most of the substrates that were identified before, they were directly associated with the proteasome. So that's the first thing that we assayed. We took a few peptides that came out in the screen, and then in collaboration with the Glickman lab from the Technion in Israel and his postdoc Ajay, we assayed some of those peptides. So we take purified proteasome and then incubate them with GFP fused peptide and then monitor the degradation in vitro. So what we were able to show that, yes, indeed, some of these peptides can directly associate with the proteasome and get degraded. So you can see the in vitro assay. A few hours in incubation, the peptides get degraded, but if we had a proteasome inhibitor, it doesn't happen. So, and this is the quantification. It can happen both if we incubate the peptides with the 26S or the 20S. And the problem is that you can see that it's pretty, it's not so rapid. It takes a few hours to get proteolyzed. So what we think is that the difference between in vitro, that it takes a little bit more time than what happens in vivo, is that there might be that there are in the in vitro assay, we are missing some assisting factors that might help degrade these C-terminal degron better. And the best indication of the best example of C-terminal degron driving ubiquitin-independent degradation is the ODC degron. And also in that case, people have shown that if you incubate the proteasome with ODC, it can very efficiently interact with the proteasome, but the degradation is slow because it needs antizyme, a cellular factor, to help it get degraded more efficiently. So we think that there might be that some cellular factor are missing in the in vitro reaction, or it might also that inside cells, the substrate are so efficiently degraded, so they are degraded even before they are fully folded. That's why it happens faster than what we see in the in vitro reaction. We have already folded proteins that are incubated with the proteasome. So that's our two assumption why it takes longer in vitro than what happens in vivo. But then we say, okay, direct association is good. It can explain some of the cases, but not all substrate were degraded in vitro. So we search for some cellular factor that may help to degrade those substrates. So using some genetic screen, we found proteins of the ubiquitin-like family that are involved. So UBL protein, there are around 50 or 60 proteins in human. And we found specifically that ubiquilin family members, there are three abundant family members, ubiquilin 1, 2, and 4. We found that if we knock out the 3 family member, now there are some substrates that cannot get degraded in the proteasome. And why? Because this protein, they have UBL domain, so it looks like ubiquitin, and it can interact with the ubiquitin receptor. And on the other side, they have protein-protein interaction domain that interact with the substrate. And that's how they bridge the proteins to the proteasome to mediate the destruction of those proteins. So shuttling factor can explain the association of some substrate of the ubiquitin-independent substrate to the proteasome. And at the same time, we published our paper. Also, the Ellis lab published a paper about midnolin and other UBL proteins that can also serve as a shuttling factors to mediate destruction. So UBL proteins, as I said, there are 50 or 60, and there might be more examples of these kind of shuttling factors that help degrade ubiquitin-independent substrate. But again, ubiquilin would not explain all the substrates that are degraded by ubiquitin-independent that we discovered. So we think there are more assisting factors that may help to degrade. It can be from the UBL family or even not from the UBL family. So we better understand now, we know more about the mechanism of targeting to the proteasome. But what about the scope of substrate? So for this, we did screen with the full collection of open reading frame. And now we don't need thousands of examples because these are fully folded proteins. And so we had a few dozens of substrate. And if we compare it to the Ellis lab screen, so the Ellis lab screen found for midnolin transcription factor that are specifically targeted by this UBL protein. Our substrates were more diverse. There was only one class. We found that either protein that are mislocalized. So with the GFP fusion, we found them to be mislocalized, and then they are degraded by ubiquilins. And some proteins were just regulatory proteins involved in cell cycle and proliferation. So it suggests overall that ubiquitin-independent mechanism can account for both quality control mechanism and also signaling on regulatory purposes. And again, all the screens were done just in normal, healthy cells. We didn't stress them. And we expect that probably if we now stress them, and some of the proteins will be mislocalized or misfolded, we might find more examples of ubiquitin-independent substrate. Okay, so to summarize this first part of the talk, we show that ubiquitin-independent degradation is much more prevalent than previously appreciated. We show that C-terminal motif drive the degradation of substrate in a ubiquitin-independent manner. We were able to map those degrons and know exactly the motif. Some ubiquitin-independent substrate can directly associate with the proteasome, while others need the help of assisting factors or shortening factors, such as the ubiquitin family members. So the question is really UB or not UB. But what are next? What are the next challenges? What are the next interests that emerge from this study? So first, think about the potential of those ubiquitin-independent degron as product, because for product, you need an E3 ligase. And in those cases, you bypass the need for E3 ligase. And I showed you, these are very short degrons. So here I just show again from our published work, some degron. So you can see that some of these degrons has the amino acid that I showed you before. They are enriched for amino acid alanine, cysteine, and valine. Some of them are just hydrophobic degrons, no specific sequence, but they are all located in the C-terminus. Forgive me. So here what we did, it's not a saturation mutagenesis experiment. In this case, we took many more examples of peptides, and then we mutate just using a single substitution in each one of the position, triple substitution or triple deletion. So you can see on top the wild type sequence, and then in red, you can see mutation that now disrupt degradation. So these are the critical residue for degradation. So you can see, again, they are all at the very C-terminus. They are very short, and they are composed just a few amino acids that are enriched for alanine, cysteine, valine, or in some cases, those hydrophobic amino acid. So if you think about product design, it needs to be either a very short peptide or some a small molecule that resembles the sequence of those peptides, and then it can efficiently bypass the need for E3 and target proteins for degradation. But for that, of course, we will need also to understand what is the targeting mechanism. We know it interacts with the proteasome, but where on the proteasome? Does it bind the ubiquitin receptor? Does it bind other subunits? So we don't know it yet. We need to collaborate with structural biologists in order to understand how. But these things are really interest us, and if there are suggestions from the audience, I'm happy to hear later in the question section. Okay, so we need now to revisit the scheme that I showed you before. There is no E3 now in this scheme, because those substrates that are terminating with alanine or valine or cysteine are degraded in a ubiquitin-independent manner. So this work was the work of Yara Makaros from my lab. And now I'm going to switch to the second project. And as I said, the second project will be more focused about biology, about terminal degron. So now I'm going to switch both to ubiquitin-dependent degradation and also to the N-terminus. So now we are going to look on N-terminal degrons. So in our 2019 paper, we found a new type of degron in the N-terminus, which is composed of glycine. So glycine in the N-terminus was efficiently degraded again by calling two substrate receptors, ZIG11B and ZER1. But then when I opened my lab, we searched for more examples on N-terminal degrons. So I took the data from our screen in this paper, and then I look for specific motifs that are enriched among unstable peptides, stable proteins, and that is the focus of this part of the talk. So when we look on this data, we found that for some reason, there are many proteins with proline in the third N-terminal position that are unstable. So this was one of the most unstable motifs that we found in the N-terminus, but we didn't know how it is degraded, which is the ligase degrading it, and why do we need to degrade proteins with proline in this position. So we started by looking first if proline is really critical for degradation. So again, I show you just one example of one protein that has proline in the third N-terminal position. And again, we do the saturation mutagenesis experiment where we mutate each amino acid across the sequence to the other 19 possible amino acid. And you can see that only mutation in the first 3 positions had an effect. From position 5 and on, nothing happens. Like the whitish color mean no change in stability. So indeed, proline is important, but not just proline. What comes before and after is also important. But again, a very short degron in the N-terminus centered on proline. Proline is critical. Now we were more careful. We wanted to make sure that it's ubiquitin-dependent degradation. So we treat some example proteins with proline with the E1 inhibitor or the proteasome inhibitor. And in both cases now, the substrate gets stabilized. So we are dealing with an E degron that is composed of a proline and degraded in ubiquitin-dependent manner in that case. Okay, so now we can search for E3 and you expect to see an E3 in a CRISPR screen. So who is the E3 ligase? We do this CRISPR screen with an E3 ligase library. And yes, we found an E3 and we found the UBR family of E3. And it makes sense because UBR are the most famous E3 ligases of N-degrons. They can recognize many amino acids at the exposed N-terminus of human proteins. And so we found UBR2 and 4. But there was some puzzling results because what we know from the literature, and those of you familiar with UBR E3 ligases, is that they recognize those amino acids that I indicated here. It can be positively charged amino acid or hydrophobic and aromatic amino acid. So none of them is not proline, and definitely not proline in the third N-terminal position. So how come UBR can recognize this type of degron? And then we looked a little bit in the literature, and then we got puzzled even more. Because if you look on one of the earliest papers, they look about how those UBR proteins recognize the substrate. So they are named UBR because they have a UBR domain. So this is the main protein-protein interaction domain. And if we look on the UBR domain here with the peptide that start with, in that case, arginine. So the Song lab crystallized the peptide, and then they tried to do some mutation. And they found actually their conclusion is that any amino acid beside proline can fit into the UBR box. So position 3 proline cannot accommodate. So now we were really puzzled. How can it be that genetically, when we knock out UBR E3 ligases, the substrate are not degraded anymore, but they cannot interact with it? So one hypothesis is UBR proteins are huge proteins. So they might utilize the UBR domain to interact with the N-terminal degron that I showed you before, but for proline, they have a different protein-protein interaction domain. So that was one assumption. But because these are huge protein, it was really hard to really assay it. So we decided to do some computational work and look on all the substrate that we have and then try maybe to better understand the motif. So look beyond the proline. So we took all the UBR substrate that we have from the screen, and then we look what comes before and after. So if we look on the amino acid that comes before proline, you can see it's a small amino acid, alanine, glycine, serine, proline, okay? doesn't say much about the degron. But now if we look on the fourth position, now we see positively charged amino acid, aromatic amino acid, hydrophobic amino acid, all the amino acid that UBR likes when they are exposed. So our hypothesis was maybe there is some cleavage events. You cleave off the first three positions, and then you can expose the fourth position for the UBR to interact with the UBR domain. So we all know that when proteins are synthesized, then methionine aminopeptides cleave off the methionine if the second amino acid is a small amino acid. And I just show you the substrate of UBR. You can see here, they have a small amino acid in the second position. So that happens in the UBR substrate that has proline. But then we say, okay, what happens next? There should be another protease in the cell that should cleave off dipeptide post-proline. So it took us some time and literature search because I Googled all options that you can imagine about peptidas, proline, and nothing comes out. And then just reading random paper, not related at all to UBR proteins, I found that actually the cytoplasm of human cell has two family members, DPP8 and DPP9. They are family members of the dipeptidyl peptidases. That this is exactly what they do. They cleave off dipeptide post-proline to expose a new N-terminus in the proteins. Previously, most of the work was concentrated on hormone and peptides, and there aren't so many full-length proteins that can be recognized by those enzymes. But what we found that this is what happens. So if we now inhibit DPP in cells using an inhibitor, or we knock out the two family members, DPP8 and 9, now we can see a very efficient stabilization of the substrate, indicating that these are regulated by TPP enzymes. So what happens is that following DPP cleavage, now we expose a preferred degron for UBRs. They can interact using the UBR domain to this N-terminal degron, and now promote the ubiquitination and degradation of the substrate in the proteasome. Okay, so we figure out the consensus of the degron. We found the E3 ligase that regulates this degron, but what's the biology behind the degron? Why do we need proteins with proline in the third position to be regulated by UBR proteins? So we say, okay, let's see what kind of protein has proline in the third position. So we ran David on the list of substrate that we had, and we found that there is a signature of signal peptide. So we saw that many of the substrate, these are secretory proteins that has proline on their signal peptide. Okay, so it still doesn't say much. Maybe proline in signal peptide helps them to get better inside the secretory proteins. Maybe it interact with SRP, the translocon, and help them translocate in the secretory pathway. It doesn't say still anything about degron. But we hypothesize that maybe proteins that have signal peptide, once you inhibit their entry into the secretory pathway, and they will remain as a cytoplasm, now they will be substrate of the UBR proteins. So we speculate that UBR play a role in protein quality control to eliminate those mislocalized secretory proteins. So what do we know about protein quality control of mislocalized proteins? We know that normally the targeting signal, if it's either the signal peptide or the transmembrane domain of membrane protein, if everything is okay, the protein is translocated across membrane and is protected because the degradation mechanism doesn't do anything to him. However, what happens if translocation fail? It can due to mutation in the targeting signal, or it can be due to interference with the translocation machinery, such as what happens during stress. So now all these secretory proteins remain in the cytoplasm. And now it's dangerous because these secretory proteins, they have either the signal peptide that is hydrophobic in most of the cases, or the transmembrane domain that now they can aggregate and the cell can suffer from toxicity. So we need some kind of quality control mechanism to deal with it. And the main quality control mechanism in eukaryotic cells, in mammalian cells, is BAG6. So BAG6 is chaperone that can recognize long hydrophobic stretches of in proteins. So it specifically interacts with those mislocalized proteins and then recruits an E3 ligase named RNF126 to mediate ubiquitination and degradation of the substrate. And however, when we look on the hydrophobicity of secretory proteins, so not all of them are hydrophobic. So we speculated there might be another parallel pathway that now will deal with non-hydrophobic signal peptides and then help them get degraded when they are mislocalized. Okay, so how do we prove it? So I just show you one example that we had in the paper, but we have many more examples. So we took one example of a protein that is located normally on the membrane. It has a transmembrane domain and a signal peptide. You can see it has a proline in the third N-terminal position. And then we created a deleted mutant in which we deleted the transmembrane domain and also part of the signal peptide in a way that it will keep the first 10 amino acid of the degron, but it won't be functional as a signal peptide. Hopefully the protein will be located in the cytoplasm. And then in order to monitor the protein in cells, to monitor the localization, we use the GPS vector. So now this protein, REL1, will be fused to GFP. We have DS-RED as a reference for the expression. We can look by microscopy about the localization, and we can also analyze by FACS the stability of the protein. So how does it look? Normal cells, wild-type cells, no treatment, it's on the membrane as expected. However, when we look on the mutant protein, we don't see it at all. So you can see that DS-RED is expressed. So we know that the whole construct is expressed in cell, but we don't see GFP, indicating that indeed this protein is degraded. So how can we restore the expression of the protein? In that case, I show an example with the DPP inhibitor, but we also have manipulation using a knockout for the UBR E3 ligases or a knockout of the DPP. So now you can see that the level in the cytoplasm in the nucleus are restored, and it's not located on the membrane. So this proves that indeed the mutant protein, what is mislocalized, it gets degraded by DPP UBR axis. So this is, again, just one example. We have more examples. So it seemed to be a more global phenomena. So if to summarize, we can see that there are two parallel pathways. One that deal with the hydrophobic degrons, the other one deal with the non-hydrophobic targeting signal. And we want to show that there is some survival advantage of having those two mechanisms acting in parallel. So we search for a good way to inhibit translocation. So now to look on endogenous protein build up in the cell as a result of inhibition of translocation. And so there is a very efficient Sec61 inhibitor. So that inhibits the Sec61 translocon. The name is apratoxin A, and we got it from the Pavilionian lab in Finland. So they shipped up the drug, and then we did some colony formation assay and survival assay. And I just show, again, just one example. So if we treat the cells with this inhibitor, they really suffer. So after 3 days, around 40 to 50% of the cell die, but you don't see a difference in survival between control knockout cells and DPP knockout or BAG6 knockout. So independent knockout of these pathways doesn't do much. However, look at the dramatic effect when we now knock out the two pathways. So if we knock out both BAG6 and DPP enzyme, now the cell really suffer because there is no any more redundancy. There is no way to compensate for each other, and the cell now suffer more from the buildup of all these mislocalized proteins. So what I showed you now in the second part of the talk, a little bit biology behind degron search. So we found a protein quality control mechanism in which the degron is located in the N-terminus, in the signal peptide. And once this, there is a cleavage event of methionine aminopeptidase and DPP enzyme, they expose the degron in order for the UBR family to degrade, to mediate degradation. And this applies for secretory proteins that are mislocalized. So the targeting signal serves both as a way to target the proteins to its correct localization, but if it fails, then it also serves as a degron. So it has dual function. And this pathway, this project was led by two very talented graduate students, Adi and Koren. And this pathway, as I said, operate in parallel to the BAG6 that mostly target the hydrophobic targeting signal. And those two pathways work on secretory proteins. So a recent paper showed that this phenomena is actually more broader. So the Rappel lab showed recently that the sci-fi complex composed of UBR and KCMF, E3 ligases, it can target mislocalized mitochondrial proteins. So if something bad happens and mitochondrial proteins remain in the cytoplasm, in that case, the mitochondrial targeting signal, again, serve as the degron that is recognized by this E3 ligase complex. So similar to secretory proteins, the targeting signal has dual function. Also here, the mitochondrial targeting signal has dual function. So we think it's a more global phenomena, and we expect to see it also in other classes of organellar proteins. And with that, I would like to finish. So this is my current lab members. And our collaborator, Richard Teams, Chao Shu, and Michael Glickman, our funding. And thank you for listening.